All right, guys, we've been talking about. Hi, guys, this is Mr. Arnold. We're going to talk a little bit in this video about nucleic acids, specifically about DNA, because obviously DNA is what carries our genetic material. And so, since we're talking about heredity, it's pretty important that we know something about DNA. So, first off, you may have had the thought in the back of your head, and you probably knew this in middle school: what is DNA? What is RNA? DNA just stands for deoxyribonucleic acid. RNA is ribonucleic acid. Obviously, deoxy going to indicate something about the oxygen in the chemical structure there. Okay, so let's dive right in. So the first thing is to figure out how we got to the point where we knew that DNA was what mattered, that was the genetic material that mattered. Back in the early 20th century, when they started investigating this stuff, there was a lot of controversy about whether or not it was protein or DNA that actually was what passed down our genes. So we've been talking about genes and genotypes and all that stuff. And so back then they didn't know how those genes got passed on. We know now, obviously, that it's chromosomes and that the DNA is in the chromosomes, but they didn't know that then. And what gave them their first big clues was something called the Hershey Chase experiment. And this experiment, this is basically where they figured out that it must be DNA that passed on the material. Now, we're not going to go into the specifics of the experiment. They put in some radioactive isotopes. They had it in the sulfur, and so they could tell the difference between the protein and the DNA. Not important to us, just that was the experiment that sort of showed them, hey, DNA must be what passes it on. In the 1920s, they start to break down and figure out that what DNA is made up of are nucleotides. Okay, that's what this word here is. And a nucleotide basically is a unit made of three things. Okay, so a DNA is what we call a polymer. There's a bunch of things linked together, and the nucleotides are the things that are linked together. And each nucleotide is made up of these three parts. So you got three things that really matter for us here in DNA that make up each nucleotide. So the first thing is a phosphate. Okay, that's this molecule right here. The second thing is some sort of sugar, and the type of sugar depends on whether it's DNA or RNA. Deoxyribose for DNA, or ribose for RNA. And then a nitrogenous base of some kind. And the nitrogenous base, I don't want to say it's the most important part, but it's kind of what gives each nucleotide its distinct what it is. So the types of nitrogenous bases, there are basically there are actually 5 total nitrogenous bases. There are 4 for DNA and there are 4 for RNA. They have 3 of them in common, and then they have one thing that's a little separate from each one. So if we just sort of number down through them, 1, 2, 3, they all have those 3, DNA and RNA. And then the fourth one is either thymine if it's DNA or uracil if it's RNA. Okay, so that switches out a little bit. When we talk about RNA, we do a transcription translation a little bit later. That'll become a little bit more important to us. For right now, we're talking about DNA, so these are our 4 bases: cytosine, guanine, adenine, and thymine. Okay, so they figured that out in the 1920s. Then this guy named Chargaff discovered this really awesome thing, which was that not only do we have 4 bases for DNA, 4 base pairs, or 4 bases, but that they always pair up in the same way. Okay, so the way it works out essentially is that adenine always pairs with thymine, and guanine always pairs with cytosine. Okay, they're always matched up in that way. So it's always the A with the T, always the G with the C. And so those things then become called base pairs because it's always AT, it's always GC. Now you can reverse these. It could be TA and it could be CG on different strands, and we'll talk about that in a second. But they're always paired in the same way. You can never have an adenine paired with a guanine. It doesn't work that way. They won't match up. They won't bond together that way. It doesn't chemically match up. That's the problem. Okay, so getting down to actually getting to the discovery part of DNA, there are several names involved. Watson and Crick, of course, is the one that we always know. But someone that gets left out all the time is Rosalind Franklin. She actually did the X-ray diffraction, which is what this picture here is, that actually showed what the general shape of DNA was, and Watson and Crick were then the guys that sort of did the calculations and figured out how this must apply to what DNA actually is. And so Watson and DNA, their proposal comes down to these 2 things. They believe that DNA has 2 outer strands that are basically 2 strands lined up next to each other, and that in those strands they've got the deoxyribose and the phosphate. Okay, so sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. And then in the middle of those strands are the bases, the nitrogenous bases. And so you've got cytosine and guanine paired together, and then you've got thymine and adenine paired together. okay? And that's repeating in a certain sort of code, and that code, of course, is what our genes are, okay? And then they believe that that was arranged in what they call a double helix. And a double helix is basically if you had all of these things arranged like a ladder, okay? So you had the sugars and the phosphates along this way, and then you had the base pairs in the middle linking it up together. You've got sort of a ladder. And then if you take that ladder and you twist it, then that forms the double helix that we always think about for DNA, okay? So nitrogenous bases in the middle, sugar phosphate backbone. And what I don't like about this diagram is that it shows that the backbone here is all one color. And remember that it's two different things. You've got sugar and phosphate sort of repeating as you go along there, okay? And you've got these four bases, cytosine, guanine, adenine, thymine. This is what we have in DNA. And this is the double helix structure, okay? Ladder, and then it's twisted around. Now, another really important thing to know about the DNA is that it's what we call, and I probably should have popped this in, the strands are what we call antiparallel, okay? And I'll write that in, might pop that in later. Now, what antiparallel means, you guys know what parallel means. Parallel means two things are running next to each other, okay? Antiparallel means that they're running next to each other, but they're going opposite directions, okay? And that's the way DNA works, is that to get it to line up the way it does, it's running anti-parallel, that one strand goes this way and the other strand is next to it, but it's running down the opposite direction because you've got the opposite base attaching on each side. So I've got an adenine here, okay, and then on the opposite strand, I've got a thymine connected. Now, the way that we talk about that in DNA is that we say that there's a 3' end and there's a 5' end. So let me just real briefly show you why, how we get to that 3' and 1' end. So if you notice here, first off, the base pair always has to be bonded to the sugar, okay? One mistake that a lot of people make is that they just sort of throw them in there. The phosphates are linking the sugars together. The sugars are then linked to the base pairs, okay? And so you see the base pair here, thymine, is linked to the sugar, and I've got it blown up over here. So this sugar, this is what we call a 5-carbon sugar, and the way that we get these 3' and 5' is that we're going to number the carbons on the sugar. You don't see the carbons. We use carbon so much in science and chemistry that we don't even really write the C anymore. We just know that each little bend here is a carbon, and so there's a carbon right there. And we're going to call that carbon, it has a name. It is the 1' carbon because that's where the connection is. And then there's a 2' carbon here, and then this carbon is the 3'. So we call this the 3' end because that's the carbon that is sticking up at this end of that sugar, okay? And then we would have a 4th carbon right here, a 4', and then a 5', and you'll notice that the 5' is still connected to a phosphate. So then if we look down at the other end of the DNA molecule, okay, and then we go through and do that same numbering scheme that we just did, okay, so we've got 1' here, and 2', and then 3', and then 4'. Then the end that is sticking off here is the 5', and so that becomes the 5' end. So this one, basically, if we look at these strands, this strand is running from 3 to 5, and then this strand is running from 5 to 3. Okay, that'll become real important to us when we talk about replication in our next video, which is the copying of DNA that happens during mitosis and meiosis that we've already discussed.